Okay, thanks very much, everybody. So I've been asked to tell you about some of the work I've been doing looking at trying to personalize the risk calculation for people at risk of MND. And hopefully we'll just talk a bit about this word penetrance that you've heard already. And then I'll just talk a bit about the genetics and what that really means in MND, and then go into how, well, can we calculate an individual's risk and think about why the risk varies. We don't know yet, but we'll think about it, and then what we're hoping to do about it. Okay, so this thing of penetrance, is a genetic term, and it's really the proportion of people who have got a specific genetic alteration who then go on to develop a condition. So if you have complete penetrance, you will always get the condition, the disease. But if you have what we call incomplete or reduced penetrance, then not everybody is going to get the disease, even though they've got the genetic predisposition. And there's a couple of examples we can show. So this is an example of a completely penetrant condition, achondroplasia. Everybody who has got achondroplastic dwarfism has got basically the same mutation, and they will always, anybody who has that mutation will always have the same condition. It never really varies. So that's completely penetrant. And an example of an incompletely penetrant condition is something called long QT syndrome. It's a cardiac condition where your heart is at risk of developing funny arrhythmias and things. And there are genetic causes of that, but even the most common genetic cause only has perhaps a 40% penetrance. So not everybody will have a sudden arrhythmia and collapse, but a proportion will. And so that risk can actually vary in a very similar range to some of the motor neuron disease genes. Another concept to think about are other terms called expressivity and pleiotropy. And you might have come across this if you've looked into this. And while the penetrance is the proportion of people who will get a condition, the expressivity is about really how severely affected is a person. And that, I suppose, might be less relevant in MND because it's always going to be a severe thing. But another term is something called pleiotropy, where you could have one specific type of condition, but you might get another related condition. And that possibly might apply if you're thinking about frontotemporal dementia versus motor neuron disease. Those are clinically different sorts of conditions, but they might have the same underlying genetic defect. So is MND actually a genetic condition? And the answer is sometimes, to some extent. And you've seen this sort of graph before, cataloguing that it doesn't project very well, but there's going to be two little lines there. And the upper one you can't see very well, but those are the genes that have been discovered over time causing MND or related to MND. And the top part is familial cases, and the bottom part is sporadic cases. And of course, we know that you can find genetic alterations in both familial cases and sporadic cases. It's more common in familial cases, but the fact that you get apparently sporadic cases with no family history tells you that it must be possible to have inherited that from a healthy parent who didn't get the disease. So there's something unusual going on here, and it's pointing towards this incomplete penetrance. So then actually, what is the chance that we're going to find a genetic mutation, a genetic alteration in motor neuron disease? And you can go and look at the literature, and we've talked about the C9 expansion before, which is the most common finding in European populations. But there are others, and you'll see here that the blue in sporadic cases is the C9orf72 expansion, and the other genes contribute less. And in familial cases, it's a similar sort of pattern, but it's less. And overall, if you count all of the cases, then actually there are a lot more people with no family history who have the C9orf72 expansion than the people you find in familial cases. So that's again telling you that is incomplete penetrance. It doesn't mean you're definitely going to get the disease if you've got the expansion. And we've had a look at this now that we have access to these genome databases and there's more and more sequencing happening than ever before. We can go and look and see how common actually are all these things in databases where people are not supposed to have MND, they're just supposed to be members of the general population. And when you do that, and you look at published cohorts as well, you can work out what you expect to find based on what we know about how common MND is, how common these different mutations are, and the split between sporadic and familial cases, and then go and look at actually how common are these in the genome data. And we find actually even for genes like SOD1, actually there's an overrepresentation of these mutations in apparently healthy population data compared to what we'd expect. And that's telling you that, again, not everyone is going to get the condition, even if they've got what is supposed to be a disease-causing mutation. So then you can go further. Now, actually, we could talk about if you picked anybody off the street, what is the chance of them having a disease-causing mutation in a gene that's supposed to cause ALS? And this varies from gene to gene. But overall, if you take all the genes that we think cause MND, then there is about a 7-fold overrepresentation of these in the population compared to what you'd expect. So that's an interesting thing, and we're still doing more work on that. But it's, again, just illustrating the point that it's really variable. And it gets even more uncertain if you include other mutations in genes that you're not sure what they do. And if you've been to the genetic clinic, you'll hopefully have been talked to by us to say that, well, if we do a genetic test, we may find something that we're not sure about. And actually, our genomes are full of things that we're unsure about. If you sequence any particular individual, you'll get about 6 million changes compared to the reference standard genome. And a lot of those, we don't really know what they do. So what is the chance of having what we call a variant of uncertain significance in one of these ALS-causing genes in any particular person? And it's actually very, very high. It's between 5 and 12%. So that's really high. Okay, and that's one reason why we don't really advocate just sequencing someone unless we know there's already something present in the family that we can go to and say, well, we know this is present, someone who was affected, we can test you for that. But if we were to test without knowing what we're looking for, there's a really high chance that we're going to find something that we really don't know what it means, and it might well be the wrong answer. So that's sort of why we do the process we do in the genetics clinic. So what I'm trying to say is that there are probably stronger genetic factors and there are weaker genetic factors. And this probably has a link into family history and how likely you are to get the disease. And actually, if you think about it, if you have a very highly penetrant condition, then you ought to really see this happening, being inherited through the family, and it ought to give you familial disease. But if you have a weak genetic factor that just increases your risk a little bit, then that's very unlikely to be passed down through the family and to show the disease in every generation. That's only going to pop up every now and then, and so that's going to appear as though it's sporadic. And so that's sort of, we could sort of potentially fit in different genes into this little picture here to say which ones seem to give you a higher risk, which ones give you a lower risk. So that was all quite population-based and wasn't really focusing on individuals. So what about individuals? Can we use what we're learning about this to try and work out an individual's risk of MND? And I guess the first place to start with this is the C9orf72 expansion because it's the most common one. And people have started to collect this sort of information. And a few years ago, there was a big analysis of many different cohorts of patients who had this to work out what ages were they when they developed either MND or FTD. And so actually that data is published and it's available and you can go and look at it. And it sort of follows this sort of curve here. And you'll see that basically after the age of 40, the risk starts to go up there and it goes up quite quickly in 50s and 60s. But then after 70s, it slows down and it's rarer to get this in extreme old age. So this is helpful information. It can sort of help us think about how much of an individual's risk has that person lived through. And if they get to 80, they've probably lived through most of the risk of getting this condition. Whereas if they're only 30, then they're just at the start, really. And we can pull out the data and look at the difference between males and females who are at risk. And it's pretty similar. There's a little bit of difference. But again, this is something that we can then apply to an individual's family history. So let's think about a family history, and if you've been to the genetics clinic, we'll have spent a lot of time hopefully talking about your family. You might be thinking, why are we talking about your family? Why are you not talking about me? But this is because the family is the best thing we've got at the moment to work out what's going on in terms of your risk. And here's a completely fake family tree, but where everybody's lived to 80, and only one person at the bottom there has been diagnosed with senile or 72-related motor neuron disease and frontotemporal dementia. So we know that this expansion, it must be inherited, and therefore we know that there's 3 people in this diagram who must carry the expansion, but only one of them has been affected. So therefore we know that the penetrance of the condition in this family is 1 out of 3, so 1/3. And you can actually work this out through just working out what's the risk of each of these individuals having the expansion. We know that the parents is a half of a risk, and then the grandparents a quarter risk that they've each got the expansion. And we know how much of their life to 80 they've lived through, and so what's the proportion of their risk they've lived through. And we know how many people are affected, and we can basically use this to weight the risk and calculate what the penetrance in this family is. And lo and behold, it comes out at 1/3, which is what we expected. So that was a bit easy, but you can go on to basically any sort of familial structure that you care to take. And this is another example where you can work it out, and you just follow where on the line of the risk people have got to in terms of their age and whether or not they're affected, combined with their risk of having an expansion, and you'll come out with a particular figure. And so in this example, at 77%. And so that's a higher risk. And what we're hoping we can get to with this sort of estimate is basically to give individuals a personalized view of their trajectory. So in that original fake pedigree, that was pedigree A, so overall the risk was 1/3, and so actually over the course of one's life, that's the sort of trajectory of risk that you're going to experience. However, if you're from that other pedigree, your risk is higher, and so you can see that over the course of one's life, your risk goes up higher. And so this is what happens in cancer genetics. People who have had that sort of experience will be shown these sorts of graphs, and that helps inform their decision. You know, what should I do? In various cancers, there's sometimes preventative surgery and things you can do. And at the moment, we haven't quite got there with MND, but that's the hope that once we have things that can actually really affect the risk of disease, people can really have a think about, well, do I need to do this yet, or should I do it later? And so that's sort of where we get to with this. Again, that was completely theoretical, but what about the real world? So we now have the ACORN study, and we have the family history data. And so we can apply this technique to the family history data. And so far, we've looked at about 52 families, and there are more waiting to be looked at. But we really see this range of penetrance based on that calculation. That goes anywhere from about 27 up to nearly 100% really. So that's a real scatter, and it's distributed in quite an even way. It's not really clustering particularly any particular bit. And actually we can look in the family histories to see, well, how many people have been reported to have MND versus FTD versus other things. And actually it is strongly weighted towards MND. That's probably just because people have been recruited mainly through the MND clinic. But there's a variety of other conditions that are in there. And you might remember that I showed you that sort of population-based penetrance was quite low. It was about 30% or so, and that was quite different from what I just showed you from the family histories. And that's probably, again, because people who are being recruited to ACORN are more likely to have family histories. And so there's a tendency for that penetrance to look higher. And probably the reality is that, well, we know from looking again at genome data that about 1 in 700 people have got the expansion, which is more than you'd expect. And again, if you look at the people who have got family histories, then of course the penetrance will appear to be higher. But if you were to include all the other people who have not been recruited and model what would it look like if we included all the people who don't have a family history, then it's more likely to come down and shift the penetrance down closer to about 1/3. And then there's probably other people out there who have never been diagnosed with anything because they don't have a family history and they're not going to get the disease. And we can guess how many people there should be like that. And you can add them into this distribution, and that pulls down the overall risk much, much lower. So, so that's an idea of modeling that we're trying to do and trying to work out what does this really mean. Of course, remember that an individual could be any of these spots here, and so it doesn't really make sense for somebody to tell you, oh, look, your risk is the same as the population, because that's just an average of everybody. But nobody is average, okay? Everybody's somewhere on the spectrum of this. Okay, so why is this happening, and why is there such a range in penetrance? And this is not known yet, but you'll hear, I think, from Jiali Gao a bit later, who's been looking at UK Biobank, and she'll explain what she's found. There are other genetic factors, which is part of the work that we want to do to find out about. One that we think we've found so far is this other gene, which on its own isn't really going to give you motor neuron disease, but it seems to be modifying your risk. Whether or not you've got the expansion, it's modifying it. And again, that leads on to something you might be able to target to affect the risk and lower it, hopefully. And there's going to be other non-genetic factors, which again are quite hard to study, but we'll try and do that. So what are we going to do? Well, as I said, very happily, MNDA have recently said that they're interested in this and that they'd like to support me to do more of this work. We're going to look at more of the ACORN study participants. There's going to be more genome data available from those families, and also, as you've heard, Beatrix's technique of looking at the long read sequencing to look at how the expansion varies. All of that is going to be analysed in light of what we can tell from the risks based on the family history data. And we're also collaborating now with the MND group in Ireland and another one in the Netherlands, in Utrecht, who've got similar sorts of cohorts that we can all put together to try and increase the scope of this and get more information. And at the same time, we're going to look at some of these large data sets to try and pull out and validate what we're finding. So those are the main aspects of what we're hoping to do in the next couple of years. In summary then, what I've tried to say is that there's variability in this. Everybody is different, and everybody with genetic forms of ALS has a different risk. And this varies from individual to individual, from family to family. At the moment, your family history is the best estimate we've got of working out an individual's risk. There are going to be other factors, and we're just starting to get a hold of some of those, but not yet something that we can apply clinically quite yet. And hopefully those are going to help us work out why some people never get MND. And if we can work that out, we can work out how to prevent it. And that's really what we'd like to try and do. So that's essentially all I wanted to say, and hopefully that's helped to catch up the time a bit. Thanks to everybody who's taken part, and to all the collaborators and colleagues. Thank you.